So last year we published that people that had a high protein intake, so the people that had a protein intake over 20%, so over 20% of calories coming from protein, compared to people that had less than 10% of calories coming from protein. They displayed a 75% increase in the risk of overall mortality and a 3- to 4-fold increase in the risk to develop cancer. By virtue of working on aging, we also work on cancer prevention. Aging, of course, the major risk factor for cancer is aging. And so one of the, whether we look at yeast aging or mouse aging and lately human aging, the genes and pathways that control longevity also seem to control DNA damage. And so we've been very interested in that aspect. And we had published a long time ago on the relationship between the TOR pathway, TOR cis kinase pathway, and protection, so the deletion of genes in this TOR pathway, which responds to amino acids and the DNA damage. And we've shown that simple cells that lack this TOR cis kinase signaling are very much longer lived. They live 2 or 3 times longer. But they're also very protected against DNA damage, not just point mutation, but also gross chromosomal rearrangements and small insertions and deletion, et cetera. And then, so we've then followed up with mouse studies and human studies. And in mice, we've actually just published that and shown that by using dietary manipulations, in this case, we use this fasting mimicking diets periodically, every couple of weeks in mice and once a month in people. But in mice, we show that by controlling this protein and amino acid signaling pathways, even periodically, we are able to reduce the cancer incidence in the mouse by about 50%. Then we also looked at, because again, proteins are controlling this TOR signaling, and also another key gene in promoting aging or aging promotion, which is called IGF-1, insulin-like growth factor 1. And because of that protein controlling this IGF-1 in TOR, we looked at the role of protein intake in mortality from all causes and from cancer in the US population. So last year we published that people that had a high protein intake, so the people that had a protein intake over 20%, so over 20% of calories coming from protein, compared to people that had less than 10% of calories coming from protein. They displayed a 75% increase in the risk of overall mortality and a 3- to 4-fold increase in the risk to develop cancer. Now, this is a relatively small population, about 6,000 people, so we cannot use that in a conclusive manner, but certainly that was remarkable that we saw a significant 3- to 4-fold increase in the chance of developing cancer for people that had the highest protein intake. In support of that is also our study from 3 years ago with this population in Ecuador that lacks the growth hormone IGF-1, that has a mutation in the growth hormone receptor IGF-1 axis. And these subjects are also turned out to be protected from cancer and also diabetes. So I think if you put it all together, also with the mouse data showing both the protein affects cancer in mice and the growth hormone IGF-1 signaling very much affects cancer in mice. I think if you put it all together, you get a pretty solid picture about the role of this protein-dependent pro-aging pathways, which involve IGF-1 and TOR cis kinase signaling, the role of this in cancer incidence in both mice and humans. Carbohydrate intake is a little bit trickier. We have described a long time ago the role of glucose in activating a second pathway, which is the RAS-PKA pathway in yeast. And then others have shown this similar effects in Drosophila and mice. The problem is that the carbohydrate intake, high carbohydrate intake, is also associated with longevity in mice, meaning that this is work by the Simpson Group in Australia and others, the low protein, high carbohydrate diet seems to be the best for longevity. Now, that doesn't mean it's low protein, high sugar diet. It's a low protein, high complex carbohydrate diet. And this is also what we saw in the population studies that I mentioned earlier. So most likely keeping sugar levels low is important, but carbohydrate intake still appears to be the best. So carbohydrates still appear to be the best source of calories. Of course, the only other option would be high fat, low carb, low protein, and both animal studies and human studies suggest that that's probably not a good way to go. I think the take-home message, if you put it all together with a lot more studies and also centenarian studies, you look at the populations from around the world that are particularly long-lived and they have very low cancer, whether it's Okinawans and some of the southern Italians, the group in Loma Linda, you get the studies support the role for a low protein, high complex carbohydrate diet with low sugar, and high healthy fats, the fats from nut consumption, olive oil consumption, and this combined with a mostly plant-based diet. This seems to be by far the most, the ideal diet to minimize cancer risk. I'm Valter Longo. I'm professor of biogerontology and biological sciences at the University of Southern California, and I'm also senior group leader at IFOM Institute. My laboratory for a number of years has been very interested in the mechanisms of cellular protection. In particular, we've been interested in the connection between nutrients and protection and the entry of a cell into a protected state. And so about 10 years ago, I started thinking about how to utilize this knowledge and this discovery that we had made many years ago, that if you starve a cell, a yeast cell, a simple cell, that cell becomes highly protected. But then we also discovered that if you express an oncogene in the same cell, now that cell becomes very sensitive. And from that came the idea of differential stress resistance, which was really about what if we starve a mouse, and that's originally what we did, and then eventually a person with their cells in a coordinated way go into this protected mode, and yet every cancer cell, by virtue of having an oncogene, disobeys, and in fact it continues on to the sensitized mode, which makes them actually more sensitive to chemotherapy rather than less sensitive. So this was a very nice way to separate the normal cells, all normal cells and all cancer cells, no matter where they were in the body.